When you have these feelings of butterflies in your stomach, that is exactly this correlation with the brain. And also the feeling of having appetite is how do the bacteria produce metabolites that then satisfy specific receptors in the brain. Also, other way around, of course, when you feel stressed, what most people then have is issues with their gut.
We are not just human. We are ecosystems shaped by trillions of microbes that influence our energy, immunity, metabolism, and even our emotions. When this ecosystem thrives, we thrive. When it falls out of balance, everything feels harder. Today's episode is a journey into that ecosystem, the gut microbiome. Joining me is Dr. Nikolaus Gasche, a microbiome scientist and co-founder of Biome Diagnostics. His work focuses on using advanced sequencing and data science to understand how the gut ecosystem influences real-world health that goes beyond trends, beyond buzzwords, and beyond the wellness noise. This is Brighter 100 from Longeny, and I am Anil K.C, your host. Dr. Nikolaus Gasche, thank you for joining, and welcome to Brighter 100.
Thank you for having me.
So tell me a little bit about why microbiome? What got you interested into microbiome?
Well, excellent question. So maybe just a little bit of a background about myself. I'm a medical doctor by training. However, I've always been more into data science and understanding complex mechanisms on a large scale. And the microbiome is actually one of those amazing things in nature where you have thousands of different species and thousands of different functions that they exhibit that need to be analyzed and need to be interpreted. And that's something that really got me motivated about the area of microbiome, because at the end of the day, it's not just about the single function of a single bacterium and the single pathway or the single vitamin that they produce, but it's more about the complex interaction between all the different types of bacteria that are in our gut, for example, and are living on our skin, etc.
So for the audience who don't know much about microbiome, tell us in simple words, what is a microbiome?
So in general, the microbiome covers everything, all the living small things, you could say. So microbiome comes from the word Greek, from micros and bios, and means more or less small living. So what we mean by that is it includes bacteria, viruses, any type of fungi, etc., that actually live together with us. So when we talk about microbiome, we can talk about the microbiome in the soil, on the ground, but it can also be the microbiome, of course, that is on our body and in our body and really plays a critical role there. And that's mainly the aspect that we think about when we talk about microbiome. We have these types of gut bacteria, viruses, etc. on our skin. We have it in our hair, on our eyes. But the most important part is we have them in our gut. And there they make up the largest part. And when we mostly talk about microbiome, we actually mean we largely focus on the gut bacteria because that's where the research started. That's where a lot has been understood so far and has been discovered. And so we really take a look at how do the gut bacteria that live in our body actually influence our health. And maybe just to put this word microbiome that might be like a little bit new into perspective, we have roughly or approximately 30 trillion cells in our body. So these are cells like our skin cells. These are cells that make up our liver, our blood system. These are really our small cells that you can see on your skin. But when you take a look at the bacteria, we have roughly 39 trillion bacteria. So we have more bacteria on our body and in our body than we actually have own body cells. And these bacteria live on our skin, but they're much, much smaller so that we can't visually see them. And when we take a look at the genome of these bacteria, the bacteria actually make up 200 times more genetic diversity or pathways that are being expressed in our human genome, which in the end, of course, means more proteins are being produced, more enzymes, more functions. And that's why, the microbiome should not be neglected, since it actually is a huge thing that lives within us and impacts our health and is living with us in kind of a symbiotic relationship.
So you just mentioned about the diversity in the microbiome within your gut, right?
Mhm.
So when you look at genetic makeup between two individuals or human
Yeah.
beings overall, the difference is very small. I mean, 98% all of us are the same
Yeah.
genetically. But
Yeah.
would you say that from a microbiome perspective, the gap is far wider as between two human beings?
There's definitely a huge diversity in the microbiome, and especially across populations. There were even studies showing that if you, for example, live with your dog and you will have a more similar microbiome than two people living across the planet who don't know each other. So living together, sharing the same environment can actually brings your microbiome closer to other
Right.
people. But of course, if you have completely different living conditions, completely different diets, this can completely change as well. There are numerous studies about inhabitants in Africa, for example, that really live according to hunter and gatherer style, where you would see completely different microbiomes than you would see in the Western world.
Right.
And additionally to that, the microbiome in these populations, they actually also shifts. It's not static in that sense. So you have microbiome shift. So if you would move across the world, your microbiome could shift as well, depending on how your environment changes. But of course, maybe we'll get into that later. But
Yeah.
one of the main influences is at the end of the day diet.
So you mentioned a lot about studies having been done. Now, for decades, medicine has treated the gut largely just as a digestive tube. What has changed in the last 10 years that made scientists rethink that it is a control hub?
So, how has microbiome research really gotten started, and how what really accelerated it? When you think about it, roughly 25 years ago, the human genome was completely opened up or was completely 100% analyzed. This was possible because there was a lot of funding available. So billions went into the research into decoding the human genome, and the methods that were used, the so-named sequencing methods, were extremely expensive, and it took roughly 10 years or so to decode the whole genome. Afterwards, genomic research was really accelerated by new sequencing machines that were able to bring the price down and increase the speed of analyzing the genome. So there are companies that are producing sequencing machines right now that can actually analyze the human genome not in 10 years for billions of dollars, but can actually do this within 24 hours for maybe a couple $100. This has been a huge technical advancement happening in the sequencing area. And through the initial research that happened with the microbiome, and then being able to sequence stool samples and allowing to analyze hundreds of different species at once, this has really accelerated that field as well. And there were some initial exciting studies that happened. Especially one of the most common cases, or that everyone remembers, is the situation where they looked at the gut microbiome of mice, and how you could say a mice that didn't have like a germ-free mice that didn't have any germs, that was then transplanted stool from an obese person, and then you had another germ-free mice that was transplanted stool from a lean person, that actually the the mice that received the obese stool actually really also got obese. So
Right.
that was the first real trigger where you saw, oh my God, the gut bacteria, they actually play a role in our weight management.
Right.
And that really got things started.
You've mentioned the fact that gut microbiome impacts your weight and all your other health aspects. So when you look at the microbiome through a systems lens, what do you see that traditional medicine misses?
So there's a reason why it's called the forgotten organ. It's a very new thing in medicine in general, the microbiome. I'm a medical doctor myself. When I studied, I did not learn about how the microbiome actually impacts different parts of health. I hope things nowadays have changed, but this is something where it just simply takes time, of course, till it gets into the clinical routine, till it gets into the traditional medicine field. But there are many areas where the microbiome supports the traditional field, and where also we're really working on making new diagnostic tests, but also new treatment methods, make them possible for clinicians in the end. We're talking here about cancer research. More and more cancer research is being done on the microbiome and how it impacts current treatments. So one of the most famous ones is cancer immunotherapy. And there, the treatment outcome is actually dependent on how well your immune system looks like. So what does it mean, or how is the immune system actually linked with the microbiome? the largest part of our immune system, roughly 70 to 80%, is located in our gut
Yeah,
and specifically in the gut wall. And that's where it interacts with the bacteria. And there they can then affect how a specific cancer treatment actually works. So these are just small parts, but there's a huge variety in almost every field in medicine. The microbiome could be used to better understand diseases, could be
right.
used to better treat diseases. And I would say it's simply, it will take time, but we're on the right track there.
Got it.
Mhm.
So, do you think the gut microbiome should be considered as an organ in its own right?
Definitely. I would say I'm a little bit biased because I'm working with the microbiome a lot, but I would definitely say that the way it impacts and the way how relevant it is, it can be considered an own organ in itself. So
Right.
there are many fascinating things about the microbiome, but in general, what it really makes so special, it's like this chaotic but still structured, diverse world living in our gut. You can imagine it more or less as a rainforest that lives within our gut and that keeps our body healthy, that also takes a look at each other and helps each others. If there's maybe one bacterium that is not available or is not existing in the gut, that doesn't mean that you will now have a list of diseases occurring. But what happens is that other bacteria, they can actually jump in. They can try to reproduce the function or the proteins that, for example, or the metabolites that the bacterium produced, try to reproduce them themselves and try to fill that gap of the missing bacterium, for example. So
Right.
that's what it makes this so interesting, is that it's not always important that XYZ specific bacterium is in the gut, but in general that you have the gut diversity that can then keep up with the different functions that are required in the gut.
We keep hearing a lot about the gut brain axis. Let's talk a little bit about the gut brain axis. How do microbes actually communicate with the brain?
That's a very exciting area of research, I would say. And I think it's one that will be more interesting in the future as well. So, how come that the gut microbiome actually communicates with the brain? What is the correlation there? And there's this specific, you could call it more or less like or I would like to call it like a highway between our brain and our gut, which is the vagus nerve.
Yeah, that,
This vagus nerve is like, it runs directly from the brain to the gut and transports signals not only from the brain to the gut, but also the other way around. So metabolites that are being produced by the gut bacteria, they can actually go from, first of all, through the blood system, but also signals through the vagus nerve can then be transported to the brain. And this is really crucial because when you have these feelings of butterflies in your stomach, that is exactly this correlation with the brain, and also the feeling of maybe having appetite is how do the bacteria produce metabolites that then satisfy specific receptors in the brain. Also, other way around, of course, when you feel stressed, what most people then have is issues with their gut. And this is mainly based on this connection in between. So in general, maybe if we look at the microbiome plays a critical role in developing or in producing hormones, for example. So what one of the main neurotransmitters that everybody knows is
right.
serotonin, that 90% of it is actually produced in the gut. And also the belief is that some of the antidepressants
Right.
that have been developed, they actually try to push the serotonin levels, that they work strongly through the gut as well.
Yeah.
But there are also other gamma aminobutyric acid is, for example, is like a neurotransmitter that actually, you could say, helps in feeling more calm and try to reduce anxiety. And this is also strongly linked with gut microbiomes or microbes actually producing it. And these neurotransmitters are crucial for the brain activity. And other way around, when your microbiome is maybe not that diverse, you could say, or is not that healthy, they might not be able to produce those types of specific neurotransmitters. And that might also then affect, of course, what signals are then transmitted through the vagus nerve to your brain and how you react to specific things in the end. Yeah. So this is a very exciting area. There are specific probiotics called psychobiotics, because what they try to do is they try to improve specific strains in the gut that can then actually improve the communication to the vagus nerve. And they actually really measure that. And they try to measure the vagus nerve activity. And see how does the specific probiotic actually change the activity, and then as well how specifically did it change the situation, for example, in these cases of depression. There are also other studies that show, of course, if you change your diet in depression. So there were studies where you do two groups. One is a placebo controlled, one is a intervention group where you look at they change your diet. Both groups, they still receive standard psychological care during the time. But the one that actually changes the diet, that has, after 12 weeks, had a huge effect on improvement of symptoms and on improvement, of course, also of the gut microbiome. The idea is that in this study, for example, they used the Mediterranean diet. And the Mediterranean diet has been one of the diets that has been associated quite well with a healthy microbiome through its content of fiber, diverse vegetables,
Diversity, yeah.
etc.
What I'm hearing you say is that there is ample evidence to show the direct relationship between the gut microbiome and your cognitive health, be it anxiety, be it depression, or even cognitive fog. You're
Yeah.
saying that connection is real.
That connection is real. You can know it's yourself when you feel anxious, maybe.
Yeah, the gut feeling.
In some cases, this leads to this vicious cycle where when you feel stressed, your gut feels stressed, your gut is not feeling well, then you're not eating well again, which then in back gives you bad signals to the brain. The brain is stressed anyway, will give bad signals to the gut again. And then it's a vicious cycle that you really need to break through it and change the diet and adapt that in that sense.
We hear a lot about serotonin, like you mentioned, 90% of it is made in the gut. What does it really mean in biological terms?
So in biological terms, so when you look at the gut, you have they're called enteroendocrine cells that are in the gut wall, more or less. So these are human cells, and they are responsible for the production of serotonin. So they produce it, but they are dependent, or their production is dependent on the response to microbial signals. So the microbiome, they produce specific signals that are then actually triggering the production of serotonin in the gut wall. And this is this interesting connection between the gut lining and the gut wall with the gut bacteria in general. So when you look at the gut, you have the gut wall, more or less. You have the mucus layer as well, which is like this kind of mucus layer, and then you have the bacteria. The bacteria feed on this mucus layer. There's one of the most famous bacteria like Akkermansia mucinifila, which is more or less, the name says it itself, mucinifila,
Yes.
mucin feel, or like mucin lover. So it really likes to feed on the mucin. This could also, you could think of this has a negative effect. No, it's good because this triggers the gut then to produce again specific mucus for the mucus layer. So actually it trims the layer and then the gut produces the mucus. So when you then take a look at different parts of the gut wall, which include specific cells, so you have immune cells there. These immune cells are also dependent on the signals that they receive from the microbiome. These can be quite different signals. This is also very active research, but there's a lot of different type of signals. One of the most famous ones that comes from the gut bacteria are the short chain fatty acids. They are mainly produced on fiber, and they are then presented to the gut wall, which includes the different parts of cells. Again, you then also have, for example, the endocrine cells that produce the hormones or neurotransmitters. Then you have the immune cells that are then also producing specific immune cells like CD4, CD8 cells that are then also important.
You've said microbes eat before we do. What do you mean by that?
So, well, the first layer when you eat the food, of course, comes into the digestive system. It goes through all the different parts. And maybe one thing just to clarify there, previously, when we talk about the microbiome, or like a decade ago or 2 decades ago, there was always this idea the microbiome only exists in the large intestine. But this is not necessarily true. There's actually, the largest part is in the large intestine, that's true, but we already have bacteria in the small intestine as well. And they already start feeding on the food that you give the bacteria. So they're extremely important in breaking up complex carbohydrates. They're extremely important in breaking up complex fibers and to then produce metabolites, but also to give this energy to your body at the end of the day and give the nutrients to your body.
Right. Talking about the gut brain axis,
Mhm.
I just wanted to understand what is that one lifestyle habit that can really disrupt the gut brain axis?
So it's an interesting question. I wouldn't say that it's always easy or it's always we always try to break things down to one single thing. In that case, with the microbiome, it's incredibly difficult to always say this is the only single thing. It's always multiple factors playing a role there. There's of course many parts that have a like really deteriorating effect on the microbiome. And that can be from having a terrible diet, eating highly processed food and low diverse food as well, to specific medications. Of course, everybody thinking about antibiotics. And also, of course, having strong infections that gut infections that drive away all the other bacteria. So there are multiple things. The worst thing that could happen to destroy the gut brain axis, if I'm honest, would be to cut the vagus nerve. That would ruin the whole highway at the end.
Right.
And that at the end of the day would of course, and actually there have been studies doing that as well, where they did that in mice to take a look at it. And that was also the first time where they really saw that this had a huge interaction.
Let's talk about dysbiosis. What does it
Yes.
actually mean? People hear the word dysbiosis everywhere, but few
Yeah.
actually understand it. Could you explain?
So dysbiosis means in general that there is an imbalance of the microbiome. So that the microbiome, we mostly try to classify the microbiome more or less in harmful and beneficial bacteria. So some that are often associated with good health and better health, and then there are also bacteria that are associated with more diseases, infections, etc., and also maybe cancer. When we talk about dysbiosis, we talk about mainly it correlates about two things. The one side is that you have this imbalance, that you have maybe many more of harmful bacteria that can actually lead to infections and you don't have that many beneficial bacteria, but it can also mean in general that you have an extremely low diversity of the gut microbiome, and low diversity often correlates more with a dysbiotic microbiome because you have less bacteria in total. It's maybe easier to get more pathogens in, because what a healthy beneficial bacteria they always do is they try to maintain a healthy microbiome. So if you have a high diversity, you most likely have many beneficial bacteria and they try to keep out the pathogens. If you have a very low diversity in your gut bacteria, this can then actually lead to having more harmful bacteria come into the intestine. And I would say the worst case of having a dysbiotic gut is, for example, the C. diff infection, Clostridium difficile infection, which is one of the diseases that has been always a lot been talked about, and also a lot of research is going into that, and also a lot of therapeutic advances are going into that on how the microbiome can actually defeat that disease. So in the case of that disease, there's one bacterium that actually pushes away all the others, one more harmful bacterium. And there's of course, in that situation, you have a very dysbiotic intestine. And what you want to do is, and how you treat the disease, there are of course different options to treat, but one of the most common ones right now is that you do a stool transplantation and you try to bring back these beneficial bacteria that then push out all the other bad bacteria, and especially the Clostridium difficile of that is causing the infection. And it can be, the interesting thing is it can be a really terrible disease that also people have died from, because at the end of the day, having no microbiome leads to a weaker immune system, leads to more infections in general, and then you die basically because you have too many infections in the end.
What you're saying is a disrupted microbial community starts affecting things like your metabolism, inflammation. But is there a clear mechanistic pathway between dysbiosis and things like weight gain, insulin resistance, or chronic fatigue? Or is it more of a correlation at this point?
Yeah, we get into the topic of correlation and causation. I would say so. We have to come from multiple sides here. Maybe just to give a brief intro to that, most studies in the microbiome area, they of course are based on correlation, where we take a look at how do specific bacteria change in the case of specific diseases. So if I would have Alzheimer's disease or diabetes, and I take a look at stool samples, I would maybe see that specific bacteria groups changed. That doesn't mean that if I would maybe add external bacteria to that group, and to try to balance it to the one of normal people, that I would then be able to, that the disease wouldn't exist anymore. So that would be the causal interaction. But
Right.
in a correlation, the disease might still continue and it would be simply a signal that you've seen. Correlation is, however, still relevant because correlation allows us to build predictive models, allows us to build diagnostic models at the end of the day, trying to understand the disease better. Causation has been shown, or there were some interesting studies in the past where specific gut bacteria, especially if were linked with metabolism, and where there's one of the most famous examples are two bacteria group, the firmicutes and the bacteroids. And what happens in that case is that these are like the two largest phyla of the microbiome. So they are really at the high level of the taxonomic rank, and they are like the top group to try to group different types of bacteria according to their taxonomic uniqueness, more or less. And the firmicutes, they're, for example, very famous for getting a lot of energy and calories out of stool samples and providing that to the body. So what has been shown in that case is that you have a higher correlation of the firmicutes compared to the other group of bacteria. The stool sample at the end that you would take would have a lower concentration of calories. That means that the calories were better extracted from the stool sample when then given to the body. And if you would have an inverse relationship where we have much less firmicutes but more bacteroids, this would then mean that you have at the end of the day more calories remaining in the stool samples, that the bacteria groups, they were not able to extract the energy that well. This, of course, here you have the causality that these bacteria extract the energy. And indirectly from there, the association was made that as the bacteria are taking more calories out of the stool sample or out of the nutrition and then providing it to the body, that would mean the body has more calories. That would mean the body has a higher probability of being, for example, overweight. And so this correlation was always somehow linked to obesity. However, it's not always as simple. So
Right.
you can't, of course, say, all right, now that you have more firmicutes, you automatically will be obese. But you have this causal relationship with how nutrients are extracted from the food and then provided to the body. But of course, there are different parts that are then playing a role there.
What you're saying is, look, there is a correlation, a strong
Yeah.
correlation between your microbiome and these various conditions, be it insulin resistance or be it obesity. But at the same point in time, we cannot pinpoint and say that this particular bacterium is what is causing this particular disease.
It's very difficult to say that.
Got it.
Because the reasoning is, and that's where we come into the idea from beforehand, is if a single bacteria maybe does not exist, then the function can be completed by another bacterium. So it's also a lot about which pathways do they exhibit, what metabolites do they produce, and these can be produced by different types of bacteria, which makes it difficult. But what we try to do is, so we always, for example, we look at specific diseases and we need to weigh them. So we take a look at how does the gut microbiome, for example, correlate with diabetes. We could never build a predictive diagnostic marker based on one single bacterium that this now exists. And that's why this disease is very probable. But it's more a correlation of maybe, depending on the disease, 20 different bacteria that play a role. There are some are elevated, some are reduced, some have a more critical role in this metabolic pathway that then leads to the disease. So you give each bacterium them also a specific grade or a specific weight, and that allows us to then have a prediction model at the end of the day with a high accuracy trying to predict this disease and how the microbiome influences. But it's very difficult that you could say only based on a single bacterium that you will definitely have, for example, this disease. There are, of course, some, you could say, like some superstar bacteria or star bacteria that are very famous, that have famous names, that are just simply more common that people know of.
And more studied, probably, right?
And more studied as well. But it's still always not only that single bacterium. is going to be relevant. Yeah. And through also now with AI and with having the opportunities to analyze a lot and having the technical opportunities, we understand much better that it's maybe really not one bacterium, but it's actually many more bacteria playing a role
Yeah,
there.
and I think going forward with technology evolving, it'll
Yeah.
be far more easier for us to start looking at causation at some point in time.
Yeah.
Right. Okay. So a lot of people use the term leaky gut, sometimes
Mm,
accurately, sometimes not. I
mm.
would love for us to separate the real science from what I call the Instagram science.
Okay, it's a good thing to talk about. So leaky gut, maybe the name already says. Where did this come from? The idea was that the gut is a wall that is not completely closed, so it can let specific nutrients through, of course, but keeps toxins and bacteria out. So it's like this selectively permeable wall at the end of the day. In the case of leaky gut, this wall is not working as it is intended to work, and there are more and more toxins actually going through that, which are then leading to more inflammation and then leading to different body-wide inflammations, more or less, and can really cause metabolic trouble. There's still a lot of research happening with leaky gut, and there was also some conflicting data on many sides that we have to be honest about, because there were studies that were showing that there of course are bacteria in the bloodstream that you can look at because they are going into the bloodstream. This is not completely true, and parts of these have been retracted as well, of this information, that they're not in there all the time, because otherwise it could also mean that you really you can have bacteria, of course, in the bloodstream, but this will be more on a very systemic inflammation basis, which is a terrible thing, which you don't want to have. And what leaky gut has also been often associated with is some autoimmune
Conditions.
component that leads to the inflammation.
What causes this gut barrier to break down? What are the biggest drivers of gut permeability? Is it stress, ultra processed foods, alcohol, low fiber, or even, as you mentioned, antibiotics?
Yeah. The main thing I mean, there are different aspects. So one of the main studies that has shown that especially like high fat diet can actually lead to a higher permeability. What is also thought of is definitely having ultra processed foods. So ultra processed meaning having foods that having more than 5 different ingredients, for example, that are really processed, industrially processed, that they then in the end cause this increase and this rise of specific toxins really going into the bloodstream. It's still, however, I would say it's still really a area of research where it's also a very new kind of a disease. When we take a look at it, we also take a look at specific bacteria that are often correlated with leaky gut and try to build predictive models from them. There's of course also other diagnostic methods for that. For example, one is that you can measure sonulin.
Sonulin, right? That's a protein.
Exactly. And it's something that also increases when the gut permeability is increased. And this has often also been associated, for example, with the disease. Yeah. It's still there are many different aspects to it. And it's also something you still need to take seriously, which is important. And it can also be really a disease that contributes to other diseases and in the end. Yeah.
Okay. So, Nicholas, just on another note, you don't have to be politically correct with us. Go with what your gut says. Okay.
Yeah.
Uh, okay. Talking about healing the gut or the gut permeability, there are endless supplements claiming to heal the gut lining. From your scientific perspective, what actually supports gut barrier integrity?
Yeah, you're not gonna like my answer, I guess. But I'm the biggest fan of really focusing on diet and not that much on the supplements that I can take, but really on the diet itself. And it can be so simple because the basic research more or less is you want to increase short chain fatty acids. That's the goal you want to increase. These are, maybe just talk shortly about them. These are metabolites being produced by the gut bacteria when they digest fibers, for example. And when these metabolites, what they do is they get to the gut wall, they trigger specific immune reactions. They strengthen your immune system. They strengthen the immune barrier. the intestinal barrier, they increase the mucus production, they go to the brain as well, they signal that. So these are like these super gut fuels that you'll always hear about. For example, butyrate is one of those that is quite
Yeah, there's
famous.
butyrate and propionate.
Yes, propionate, for example, as well. And what you want to do is in that case is you really want to try to increase short chain fatty acids. So there have been also studies where they injected short chain fatty acids in mice, and that improved again the intestinal health and so on. So you can really, you know, that these have a really significant effect. And how to really increase it is mainly by focusing on the diet and trying to. So we do different types of challenges, fun projects as well. Simply try to go for a week and try to get as many different vegetables and fibers in as you can find, and try to add like 30 different vegetables in your diet plan in a week. If you think about it, at the end of the day, it's not that easy, but it's definitely doable. And this will help you because it will feed many different bacteria then being able to produce these short chain fatty acids. And they will then increase the diversity further, because if you feed many different bacteria, they will grow, and it will have a constantly feeding cycle that it will improve your gut health, your immune system. It will improve so many different things about your body. And that's why we're also a big fan of really focusing on the diet. And
Yeah. So in
the idea
fact,
is also maybe just one thing there is one of the things, because we mentioned just leaky gut as well, that has also been associated with antibiotic use, because then there's also again a dysbiotic intestine, etc. But in that case, you even if you have a very strong diversity and you then take an antibiotic, the probability that you will afterwards, depending of course on the antibiotic, that you will have a completely deteriorated microbiome will be much lower if you already have a very strong microbiome that can handle the stress of then going through an antibiotic therapy, going through different types of therapies. So that's where we see the focus. Maybe going back to your question about the supplements, of course, in some cases it might make sense, and if it works for people, we don't want to discourage them, because there are definitely very well researched probiotics out there, but our bigger focus would be on the prebiotic side than the probiotic side.
Yeah, I'm going to come to those, the pre and the pro. It's interesting what you just told me, because I was going to ask you about the whole 30 vegetables or 30 different types of greens and vegetables in a week, whether that was just hype or whether it's a reality. So what you're saying is, look, it's a reality and it does help. That's great to know. Talking about short chain fatty acids like butyrate and propionate, how do they link the gut to GLP-1 signaling and insulin sensitivity?
That's also a very good question. So you have the short chain fatty acids, and this has been, I would say, a little bit more research now happening, because GLP-1 has been, I think, quite internationally a quite well discussed topic with the new treatment methods that are out there. So what the short chain fatty acids can do as well is that what they then do is they're being produced in the colon, and they then trigger the release of specific hormones. And one of these hormones is also GLP-1. And GLP-1 then at the end is really important, because we all know it improves the insulin secretion at the end of the day, and suppresses appetite. And the same thing is that can be triggered by having more short chain fatty acids. So you can more or less, by feeding your microbiome, you can trigger your own GLP-1 at the end of the day, so that you don't necessarily need to take this medication, but you can use it your own. There have been, I mentioned before, there has been a study that in mice, I think it was, but where they actually they measured that. They took propionate, they infused it into the colon, and they really saw an increase. Then there are also other peptides that are really important for insulin secretion, but one of them was also GLP-1 that was then afterwards increased, and leading also then to the mice eating less than the ones that have not been infused with propionate.
So we now have GLP-1 drugs dominating the metabolic conversation. But do you see the gut microbiome based interventions as a parallel or complementary path in the future?
Definitely. I mean, in general, as I just mentioned, like the gut microbiome in that case is crucially important for. So I would. It's always easy to think about taking, of course, medications and treatments, but at the end of the day, the question is, can't you just maybe take a little bit more effort and then manage to do it on your own? You know, because your body is already in that case, magical thing that has GLP-1 receptors, that has the GLP-1 production. I mean, this is not something that companies came up with that you can produce, or that they found GLP-1 that this is a unique marker, but GLP-1 is something that is already being produced in the body. So
Yes.
why won't we try something out that actually might increase the production of that by ourselves? Know what I mean?
Right. We spoke a little bit about
Mm.
diets and so on. We'll get into more of it soon. But what's the one misconception about gut friendly diets you love to correct?
One misconception about gut-friendly diets. It's a good question, because there's of course this trend nowadays that you want to eat as healthy as possible. At least in many Western countries, it's extremely trendy to be trying to optimize on every side of things, and as well on diet. But at the end of the day, it's still about the diversity. So it's also fine to, of course, not trying to eat, I don't know, over 30 grams of fiber every single day. But you can, of course, have, as long as you have diverse meals and that they include a specific percentage of vegetables, in general, it is extremely beneficial for the gut bacteria.
Yeah. Okay. So when we talk about metabolism, we are not just talking about calories in, calories out. We are talking about microbial balance, barrier integrity, inflammation, the whole system. Which brings me to another area that fascinates me: inflammation, immunity, and resilience. You had mentioned 70% of our immune cells sit near the gut.
Mm.
Why is that evolutionarily important?
Well, eh, maybe there's a nice thing to say about that in general, that I like to always think about when I think about the gut. So why is the immune system mostly located in the gut? When we look at the gut, the amazing thing is that the gut has the largest surface of our body. And this makes completely sense that then the immune system is located in the gut wall. Everybody, or mostly, you would think about the skin. Also, we know this, of course, in summer when the sun is quite hot, you put on a lot of sunscreen. However, the skin at the end of the day makes up 2 square meters roughly of our body. And the gut, when you would lie it flat and would spread it out completely, there are also varying numbers, but I like the larger numbers because it resembles a sport that I like quite well. It's as big as a tennis court. So
Right.
it's a huge surface that you would see
Correct.
at the end of the day, and that is in contact with the outer world. And so what then happens is that through this huge surface, through the nutrients that you or the food that you eat, you of course get a lot of germs, you get a lot of different types of pathogens, and the immune system, and since also our microbiome is located in the gut, the immune system needs to know which bacteria are actually good and are beneficial, and which bacteria are actually the ones that are harmful, and the ones the immune system should actually work against. And this is a huge learning part of our body and our immune system, that it knows which microbiomes they should not attack and which one they should. And the microbiome also helps the immune system in that case, because it sends the immune system specific signals to understand and present also specific pathogens, to understand, okay, hey, these are bad guys. This one, the next time you see them in your body, you need to kick them
Right.
out, and you need to, that's an infection. And the other ones, they're fine. They're the good guys. It completely makes sense that that 70 to 80% of our immune systems are located in the gut. And if I can add something there, because we talked before on cancer treatment, and I think it's a good thing to think about this a little bit more. There's this new cancer treatment method called immunotherapy, which can be used in a various types of cancers, such as melanoma, but also lung cancer, renal cancer, different types of cancer in the body. It can be used. It's a quite new type of cancer treatment, so it's been around now for the past you could say, past 10 years roughly. And the amazing thing on how it works is, and this is really unique to it, is that a tumor in general or cancer in general, it grows because it can hide from our immune system. And it can grow bigger, and it tricks our immune system in thinking it's not growing, it's not there, it's a healthy cell, everything's fine. And it more or less hides from our immune system. And what the immunotherapy treatment does is the only thing it more or less does, it makes the tumor visible again so that the immune system recognizes, hey, this is a bad cancer cell. This is something we need to remove from our body. And the treatment, if it works, if immunotherapy works, patients can really get cured from it. So really late stage cancer patients in, for example, melanoma, which 10 years ago, it was more or less a death sentence, nowadays can be cured from that disease, which is amazing. And that's the one side, it can be extremely useful treatment. But on the other side, you have many cases where the treatment doesn't work at all, where it can actually lead to side effects. And the interesting part there is that what is the reasoning behind it is that even if the tumor is made visible to the body's immune system, but the immune system is maybe not that fit, that healthy, that trained, it can actually not really attack the cancer, cure the cancer, destroy the cancer. And those are then the cases where the patient might not respond. And we've actually took a look at that as well, because the microbiome, you can really see clear patterns between the microbiome and how well you would respond to the cancer treatment, because you look at the immune system. You look at how well does the immune system actually look like when you take a look at the gut bacteria. And going a step further, now that you know the gut bacteria actually play a role in training the immune system, you could actually think about maybe the gut bacteria, if you alter them, maybe the cancer treatment will actually work better. And that's something that has been done now. That what they did is a very, very, very interesting method. They took stool samples from previous cancer patients that responded excellent to the treatment and that are cured from the cancer. So they have a really good microbiome. They took their stool samples and transplanted those stool samples to recipients who also got the cancer treatment, but did not respond to the cancer treatment. So these were non-responders. The cancer was continuously growing and the immune system was not able to fight it. They received the stool sample from previous responders with the healthy microbiome, and that actually led to people who have not responded, where the cancer has grown, to then respond to the treatment and getting a reduction in the cancer size, etc. These are, of course, so this has been going on for the past 5 years now, because the cancer treatment is still new. The microbiome research to it is still new, but I believe it's really amazing to see the powerful effect of the gut bacteria, that if you transplant it, they actually alter the microbiome inside and then alter the immune system in such a way that the immune system can then, or is maybe the gut cells, they're triggered to produce new immune cells. The mechanisms are not completely clear, but that there's a strengthening happening that then results in fighting against the cancer. And that, I believe, that's one of the most amazing things.
It is. It's simply wow. Okay. We spoke about immunity. How does the microbiome influence chronic inflammation, the slow fire behind many, many diseases?
Well, that's of course also again a topic that is linked with the immune system, because it's not everything is always black and white, you would say. You know, you don't always have a high diversity, or otherwise you have a very low diversity, but you can also be somewhere in between. And this somewhere in between could then of course also mean that you have a microbiome that impacts your immune system in a partially positive way, but maybe not strengthening it in such a way that this can actually lead to common regular colds, regular different types of infections that come through the gut, but also through other pathogens. In that case, and that's where we're always going to come back to. So we're always going to talk about fiber again and the production of short chain fatty acids, because this in the end means that maybe there is some possibility for diversity in the end. There's some possibility for an increase of specific bacteria strains that need to be grown, that need to be fed in the microbiome. And there's also, at the end of the day, I don't want to be talking like everything happens from the microbiome. I think this is important. Microbiome is always just one side of the things. There's, of course, so if we take a look at, for example, chronic inflammation in the gut, is the the inflammatory bowel diseases like Crohn's disease, ulcerative colitis. These are terrible diseases, and the microbiome plays a role in these diseases. There are specific types of bacteria, specific, for example, E. coli types that are associated with the disease. But on the other side, even other patients where maybe they still have these bacteria, they don't have colitis or ulcerative colitis. And so it's always a little bit, there are multiple factors playing a role. The genome plays a role, the human genome as well, and the environment in general, in that you've grown up and then you've been living. So there always, you can imagine it as this multifactorial triangle where you have your own DNA from your ancestry, you have your microbial DNA that is mainly based on how you eat and how you've grown, and then of course, one of the main factors is the environment. Where are you living? What toxins are you exposed to? And what type of lifestyle are you living, et cetera, et cetera. So it's always important to keep these three
Right.
parts in mind, because we're of course talking about the microbiome, but the microbiome can't do the job of everything. And we shouldn't forget about the other aspects of life.
We've spoken about the fact that there is no one single bacteria that we can hold responsible for any particular disease. But in your experience, what signals that someone's immune gut balance is off even before the disease shows up?
Also, an excellent question again. For me, completely clear is the number one thing to look at is the diversity. That's the number one signal that we look at also in every microbiome report. How diverse is the patient? That gives you already a very strong idea of how well is the gut health situated, or how well is the microbial health looks like. And a more diverse microbiome, I know there have been many studies, of course, that if you have a high diversity, you can still have diseases, of course, because you always have different aspects. But generally, higher diversity always means a more stable microbiome, means more resilience to other diseases, means more resilience towards maybe antibiotic treatment, etc. There are these great bacteria, and there are very important ones like Akkermansia, for example, that is extremely beneficial and has been researched a lot. But for other diseases, we often see it's then not the impact of, or there's, you couldn't say it's this marker that you have this one bacterium, but it's more this combination of multiple things going wrong and then leading to this. And that's what also makes this so interesting, this microbiome area. We look at, if we take one stool sample, we look at, let's say, in a stool sample, you have 600 different species that you take a look at. And you list them down like you would you list them on a blood, on a sheet of paper, like you would in a blood report. A doctor would have no idea what to do with this information. And nobody would, not even I would. I couldn't even tell you, okay, now you have this single bacterium seems to be elevated or reduced. That's obvious you have this disease. It's unfortunately it's not possible because it's such a complex world. So as I mentioned before, if we look at specific diseases, we see mostly like 20 bacteria playing at least a role. Some diseases with 50 bacteria playing a role. And these bacteria are then interacting together, and there are multiple things going wrong. It's not even one single one being, for example, reduced. It can be another bacterium being elevated, another two bacterium being reduced, and another five ones where they're harmful, they're being elevated, and then another two ones that are being beneficial that are reduced. So it's always this combination of different types of more harmful and beneficial, where there's just like have this balance, and then you have this tipping point where just at some point it tips into the wrong direction.
So, you did mention a little bit about growing up with various microbes. How closely linked are early life microbes and long term immunity?
Yeah, that's also a good good question. This has been a huge part of research in the microbial world. So, for example, one of the main aspects has been starting with pregnancy and how the child was given birth. They're different. Of course,
Yeah.
you can do a C-section or you do the natural way through the vaginal canal.
Sorry, I'll just interrupt you there. I think, isn't it true that our first microbes come from the moment we are born? They actually come
Mm.
from the mother.
Yeah. And that's it depends. If you have a C-section, they might come from the hands of the nurse that actually gets out the child. So generally that's true. But there has been also shown that if you have, for example, if you get a C-section in that case, you would get in touch, or the microbiome initially afterwards that you would have is more closely correlated to the microbiome that you have on the skin than normally in the gut. Whereas if you go the natural way, you get in touch with any vaginal fluid, you get in touch also with stool fluid and all of that. And can this then of course gives you a completely different microbiome. And there are now also what is nowadays done, I'm not sure how often it is done in India, if I'm honest, but I know at least in Austria and in Europe it's very common that if you have a C-section that you
Swap the
take
baby.
a wipe from the mother and you wipe it across smeared across the face of the child after birth, after a C-section, so that
Right.
it initially gets in contact with the right type of microbiome.
So there have been experiments where researchers did swab C-section babies with vaginal or placental microbiota. What did
Mhm.
those experiments really show?
I mean, I'm not completely familiar now with the study results in the end, but what I imagine is that they, of course, they get the right type of bacteria, and they get, as far as I remember it, it was that they have a more similar type of microbiome after birth as they would have when they did the standard natural way of birth. So, which you, at the end of the day, you want to have that. Maybe just going into children and microbiome, you have, this is talking about maybe the stability of the microbiome
Yeah,
and
in fact, I was going to ask you, we
yeah.
talked about babies. So how strong is the connection between the microbiome a child develops in the first few years and their long-term immune resilience?
So maybe so in general, taking a look at the microbiome of children is extremely difficult. Why is it difficult? Because they have in the beginning almost none, and they are growing like crazy afterwards, and they grow in all directions. If you try to plot the microbiome of children on a two-dimensional scale, and you have plot them in the room like as a dot, this is the microbiome of child A, of child B, of child C, and you're going to see over the years, over the first couple of years, the microbes are going to jump around like crazy. You will have completely different species after birth, completely different species a year later, 2 years later, 5 years later, etc. So you have this huge variance that is happening. However, you almost always have this type of basic microbiome, you could say, or like basic type which you mostly tend to, which at the end of the day, we then call it enterotyping, which means that we try that when you're also then an adult, you can group the microbiome according to these three enterotypes. And these enterotypes are also strongly linked on um, the basic microbiome that you had as a child. But what is really important to say there that is that the microbiome in the beginning is incredibly like going all over the place. And then at some point it stabilizes. It also depends on the living conditions. A microbiome of a child, probably in Burkina Faso, will look completely different than the one in Italy. And it will stabilize at different points of time, saying that also I would say in rural African villages, for example, you have this variance still also as an adult because your diet changes seasonally. In the Western world, the microbiome will stay quite stable. And then as you get older, you reach, like at the beginning, you have a very low diversity. And then as you get older, you will then receive a stable, high diverse microbiome over your 20s, 30s, 40s. And then at the end of the lifetime, it will also go down again. There's also one less interesting part about it, that people who get very old, over 100 years of age, they have been associated to not have this dip at the end, that where the diversity goes down, but to continuously have you could imagine more a juvenile microbiome that has a high diversity, etc.
So what you're saying is there is a connection between longevity and the diversity of gut
Yeah.
microbiome.
Yeah, definitely. Definitely. I mean, it's interesting because, for example, the studies they did on longevity, they've been mostly done in Japan, of course, because there we have a lot of data on people who are over 100 years old. We have the data over generations, so we know how do their children, great grandchildren, etc., how do they look like, and they can compare the microbiome in that case. And what they see is that these microbiomes of the older people, that compared to our standard microbiome data that we have, they have quite a high diversity towards the end, even though what you would expect in our life after 60, 70, it dips down. It dips
Right.
down strongly. And this can also then, of course, be associated with more diseases happening, etc. So having a strong microbiome in age and having a diverse microbiome can actually be extremely beneficial and be also a strong aspect for longevity.
Yeah, I think I'm just digressing a little bit, but there's also a correlation between the Amish tribe
Mm.
where
Mm-hmm.
they don't interact with any sort of, I mean, the children go to the farms, they're in the barn houses and so on, and they
Mm.
interact a lot with animals. They have extremely high immunity, right?
Mm.
They rarely fall sick and so on. So it's got something to do with the microbiome.
I can imagine that they produce a lot of antioxidants and also some anti-inflammatory
Right.
metabolites or compounds that can then, aging, you always have this chronic inflammatory process that this is somehow reduced then.
I think the more sanitized world we live in, the lesser the diversity of microbiome.
Yeah, definitely.
Tell us a little bit about the microbiome test. Does the test actually measure? What's happening behind the scenes once someone mails the sample?
Sure. We saw this strong need to bring the microbiome from the lab bench to the clinical routine to make this analysis as scientifically based as possible and to bring this to make it available more or less to everyone. We've known the pain that there are these sequencing methods out there where you can analyze a lot of bacteria, but at the end of the day, if you get the list of bacteria on a sheet of paper, you're not going to know um, what is that really happening in your body. Nobody's going to understand it. And so what we did is we've built one of the largest databases of microbiome data worldwide, which includes the clinical profile as well. So we know quite well how does the microbiome, for example, look like of people who have, we mentioned leaky gut, but who have like irritable bowel syndrome, who take specific medications, who have a vegan diet, who eat more fish, who have specific allergies, etc. Also gender, ages, and so on. This database really allows us to understand the microbiome better and to build a lot of models on predictive models for diseases.
How accurate are microbiome scores or diversity in predicting real world outcomes?
We can call them microbiome indexes, for example, these predictive indexes. I would say that diversity is not a predictive index for a specific disease or something. So this is really a very, very informational and descriptive result that is important to look at and is always important to know how the score looks like for this specific patient. For the indexes, we have different types of accuracies. And different types of scores there. So, of course, not everyone's going to be as same as accurate, but we try to always aim for the highest possible accuracy. What often happens in the machine learning world, you train the data on some training data, then you put it live, and then you're going to get completely different results. And that's not what
Right.
we're going to do. What we're doing, we're really trying to constantly retrain. We're looking at how does the model perform on real life data, and then try to improve that. But I can tell you that in some cases, the models are extremely good, that for specific diseases, they might have accuracies or sensitivities up to 85, 90% in some, which is very good and can actually really have, compared to other diagnostic tests, a very, very strong effect. On other sides, we've actually built some fun markers as well. We try to, for example, predict the gender based on the microbiome.
For a layperson, when does it make sense to test? And when does it just feel like it's curiosity?
Yeah. So I would say there are three different types of people that do the test. The one person, as you mentioned, is curiosity. I would call them also like the biohackers,
Right.
optimizers, etc., that really want to see what is going on. They've done DNA tests. They want to figure out how does actually the microbiome affect my health? Does it influence my performance? In general, my psychological and physical health, and they want to look at that. That's the one group. And the other group are those who have active issues right now, mostly gut issues, where, as we talked about leaky gut, but we talked also shortly about irritable bowel syndrome.
Right.
These are diseases that are non-lethal, but are very difficult to diagnose, and people are mostly left alone with the disease. So they've been wandering from doctor to doctor. They have a lot of pain. Well, it has a very terrible impact on the quality of life. And There we can take a look at these diseases, see if actually the reason or aspects leading to this disease is actually coming from the gut microbiome. This is the second group that wants to figure out, does changing maybe then the microbiome really affect the disease? So getting some understanding there, and also maybe looking at it during flare-ups or remissions, etc. And then the third group is definitely preventive health, seeing where you have your, your initial weaknesses in your body, when you realize them, that you want to see, how are there maybe more things coming up where I need to improve? Are there metabolic issues coming up, metabolic syndrome, diabetes, different types of, you could say, metabolic diseases that are all associated with the microbiome, that have not occurred yet, but the microbiome already shows a tendency in that direction. For that, it can also be extremely helpful. They can be in the 30s wanting to optimize everything, but we also have in the 50s, 60s wanting to adapt the microbiome preventively.
One key aspect which many people struggle with, especially in India. Tell us a little bit about the connection between microbiome and diseases like IBS, IBD, and even H. pylori.
There are three different diseases, completely different diseases. So irritable bowel syndrome is a non-lethal disease that has terrible effect on the lifestyle, where there's no known disease or no known cause for the disease, unfortunately. It's a disease that is diagnosed based on excluding all other harmful diseases. And this is really terrible, because for the patients, they know the doctor tells them everything's all right, blood work is all right, colonoscopy is all right, everything looks good, but they still have those serious gut issues. And there's still some theories of different aspects that might influence it. There are strong theories in the direction, for example, of biofilms, and also strong theory in the direction of microbiome, that specific microbiomes actually can promote more or less this disease. Um, in Helicobacter pylori disease, I would say one of the famous bacteria that are of course this like
Yeah.
associated with a disease, they with a very famous one, and it's gastritis in that case, especially the bacterial gastritis. There are different types of gastritis, and this bacterium is associated with bacterial gastritis. There are different diagnostic and treatment options for that. It is possible to also identify it with the stool sample, but it's not really the gold standard to do that. So we don't do any predictions models for H. pylori, because it's just simply, there are good diagnostic standards for it and also good therapeutic measures. And it's also located in the stomach. So stomach is more or less seen as, or has always been seen as, more sterile than the other parts. I would
Right.
say more sterile, because it's not 100% sterile. And the H. pylori is one of the bacteria that thrives in this area, and that has specific enzymes to actually protect itself against the acid. That's why also one of the main therapeutic options besides antibiotic is also having proton pump inhibitors, which then change the acidity of the stomach, or like increases the pH value to then reduce the occurrence of H. pylori, because it then is a different environment where it can't thrive as much. And with the inflammatory bowel diseases, this is a very complex disease, also depending on ulcerative colitis or Crohn's disease. Because, for example, in ulcerative colitis, one example with the microbiome was that there's one bacterium called E. coli Nissl, which was seen as one of the missing links in the microbiome of ulcerative colitis patients. And there was then even a probiotic developed based on that. And this probiotic seemed to have a very beneficial effect on ulcerative colitis, because it filled this small gap of a bacterium that was missing in these patients. And this worked, and it improved the symptoms, but it's still, of course, it doesn't work 100%. And that's where I want to get into the topic again. This is especially inflammatory bowel disease. These are diseases that are multifactorial.
Is there a way in which we can look at gut microbiome and identify the problem for IBD?
So it depends a little bit on the disease in general. So IBD is linked to the microbiome, and the microbiome can be one linked to decode the disease, you could say. But it's always this three-part pyramid that I mentioned before. You have the human genome, the microbiome, and the environment, and we can look at the microbiome in that case. For irritable bowel syndrome, IBS, This is a disease where there's no other diagnostic option available. So the disease is diagnosed actually by excluding all other diseases. And the microbiome has been strongly associated with it. Even with biofilms, they have been associated with the occurrence of, for example, IBS.
Let's talk a little bit about diet and dietary habits. Prebiotics and probiotics. What's the difference and do they really matter?
So when we talk about prebiotics, we mean like the fibers that we talk about the food that we want to give the bacteria. So these are normal nutrients that we give to the bacteria that they can grow and they can get more diverse. This can be in a natural diet. This can also be as form of a supplement with specific fibers that are maybe easier digestible. With probiotics, we mean living bacteria. So these can be combinations of different types of bacteria, single strains, multiple strains. It really depends a little bit, where the idea is that you give the living bacteria to your intestine, that they will then grow there and continue to grow there and settle down, be part of a normal gut flora in the end.
So when it comes to prebiotics, prebiotics are everywhere now, whether in the form of powders, gummies, fortified foods. But do you think we are overdoing it? Can prebiotics cause real harm when used without understanding the underlying microbiome?
I would believe that probiotics can cause a greater harm than prebiotics.
How so?
So, for example, probiotics are quite common in the case of antibiotic therapy to do it in parallel, because the idea is you restore your microbiome. But the thing is, with the probiotic, you always have like one strain, two strains, maybe 5 or 10 strains. But the microbiome is so much more. The microbiome has 500, 600 different strains. And so if you fuel your body with the probiotic with 5 strains, they will take place in the microbiome and maybe push away other species that you actually want to keep. And so this is an issue, especially with antibiotic therapy. It has been seen that the antibiotics, they remove parts of the bacteria that would naturally grow back if you feed them. But if you instead give probiotic, they replace the bacteria that should actually be there. And this can then actually delay the process of building up a strong diversity again. Whereas with the prebiotic, the only thing that really what people are a little bit suspicious or anxious about in that case is that the prebiotics, they always think about bloating and a painful belly, for example, because what happens is that the bacteria, they produce gases based on the fiber, and that can cause pain. But here it's really important to note there are different types of fibers. There are fibers that cause these gases and that can be painful, but there are also fibers that are much more easier on the digestive system and on the microbiome, but are still very, very helpful. So there are also specific fibers, for example, for IBS patients who already have quite a bit of gut pain.
So when people think of probiotic, they simply think of just popping in a pill and fix their gut. But from a scientific perspective, are probiotics genuinely effective or have they become like the new multivitamin, expensive plus placebos with great marketing?
I'm always a fan. If I would look at a probiotic, I would look at more a single strain probiotic that is well researched. Then I would take up the next one. All the others are more or less, as you mentioned, have a kind of a vitamin effect. Although vitamins in some cases are also important. So,
Yes.
yeah.
We hear a lot about gut cleanses, colon detoxes, and 7-day gut reset programs claiming to flush out toxins. Is there any biological reality to this, or are people simply harming the microbiome by doing these things?
Yeah, it's we know about that. I think it depends a little bit on the situation, but largely it's not necessary in that sense. You already have this base of microbiome. You should nourish it and treat it right instead of trying to clean it out, because what you do is you always clean out all the beneficial bacteria as well. And the only thing that I would really look at, if it's possible in your region, of course, and if you have a specific disease or a study where this is happening, is stool transplantations, where in that case you are then able to receive more beneficial microbiome profiles from other ones. And stool transplants are very well regulated as well, in the case that you always need to do thorough testing on stool samples before you transplant it. Because at the end of the day, you can't just easily take a stool sample from somebody else and transplant it. You need to really analyze it. Are there any pathogens in there? Are there any viruses in there, et cetera, to at the end of the day be eligible for transplantation.
What do you think of keto, carnivore, intermittent fasting extremes? These are hugely popular, but restriction often means less fiber and less diversity. Are these diets, especially in the long term, actually damaging people's guts?
I guess it depends a little bit on the diet. As I mentioned, important, diverse diet. That's the most important thing. Intermittent fasting is not really a diet by itself, but what it does is that it gives your intestine a time to digest. And I have to say, I'm a big fan of intermittent fasting. I do it myself as well, because I feel very good with it. You
So
know, I
what, you
feel
do like 16-hour window or?
exactly. I mostly I have no breakfast.
Yeah.
And my first food of the day is then the lunch. And this helps me to feel already energized in the morning. Gut is getting hungry and that it's getting appetite. Whereas if I would be constantly probably feeding myself, the gut wouldn't have the break time to work on its things, you know? And I mean, there's also a lot of research happening there, but I believe that it's actually something that can be quite healthy. And then additionally, of course, we should look at fermented foods is something that should be included in every diet, and it can be as easy as doing it at home. There's a lot of things that fall under fermented food. What I love to do is bake a sourdough bread. It's quite nice to do, quite meditative to do it, and it's an amazing feeling and experience when you pull out that fresh baked loaf out of the oven, and it just smells really nice.
So most of the gut microbiome data is from the Western populations. How
Mm.
different do you think of the Indian or Asian microbiome is?
I think in general, I would say the region doesn't play such a role. You can have quite similar microbiomes across different regions. However, this only applies to if the diet is similar. So the diet really has one of the largest impacts. It's one of the largest confounding factors. If I would be living in Japan, having the same diet as in Austria, my microbiome wouldn't change as much. How does the microbiome change? How long does it take? And also retesting, because that's often a topic. We see that the microbiome is, of course, the stool consistency changes on a day by day basis, but it's not like that the stool consistency is mainly derived by the microbiomes. Changing your diet and then changing really your microbiome, this normally would require a couple of weeks to months that you really see significant long-term effects. We normally recommend retesting after 4 months, because then if you've been continuously changing something in your diet, then is it where you're really seeing the impact.
Okay. And how long does it take for somebody to reconstitute their gut microbiome from poor gut microbiome to diverse and rich gut microbiome?
That's a good question. Depends on how strong you do the intervention. If you start off with a stool transplant, it can happen quite quickly. If you do it simply by diet, but you're really into it, it will take some time, but you can definitely get there. And I think also retesting the microbiome gives you additional incentive to see the progress, to track the
Right.
progress, see how has the diversity really changed? How many new bacteria have I won since the last analysis, for example?
Okay. So if every person understood just one truth about the gut microbiome, what would you want that to be?
I would like to that everybody gets the feeling of how amazing that is, that actually our body consists of more foreign cells than we have our own body cells. And this is something where I think this is the question everybody can ask themselves: what makes me a human in the end? Maybe those are actually also all the bacteria living in myself and in my gut, because they make up more about me than I am myself. And so they influence our mental health, they influence so many different aspects. And if everybody can reflect on that aspect, then it's much easier to think about how do I want to change my lifestyle, etc. Because I have this additional thing in like growing on me in me that is actually really important for me and is
Right.
more a symbiotic relationship.
I'm mindful of your time. So as we come to the end, I'll just ask you rapid fire questions. This is a
Mhm.
tradition at Brighter 100. If you had to bust one myth about gut health that drives you crazy, what would it be?
One myth about gut health that drives me crazy. I mean, you've probably heard it through the podcast. Probiotics are not like the all healthy source that heals all problems, but more focus should be on the food.
If you could change one thing in how people live or eat to support the microbiome, what would it be?
Maybe one aspect that we didn't talk about that much is exercise. Having exercise strengthens, of course, the immune system and the microbiome as well. So including exercise is something extremely beneficial. And I would say, adding to that, what every doctor recommends every patient who has heart disease is to get a dog. And I think this actually can also be applied to the microbiome, because getting a dog means you get some exchange with microbiome from the dog, but also you get your exercise. You have to go out every day a couple of times, and you get fresh air. So that's a big plus for the microbiome.
Dr. Nikolaus Gasche, thank you for making the unseen world inside us a little clearer and a lot more fascinating.
Thank you very much for having me, Anil. It was really a pleasure talking about the microbiome, and I'm happy to join anytime soon.
To our listeners, today's conversation reminded us of something simple but profound, that we aren't just a body, we are an ecosystem. Every meal, every night of sleep, every moment of stress, it all becomes part of that ecosystem. And the more we understand it, the better we can live, not through hacks or shortcuts, but through awareness and intention. Thank you for listening. Stay curious, stay consistent. As always, stay brighter. This is Brighter 100 from Longeny.